Kia ora koutou, ko Sue Brewster, taka wingoa. Well, welcome everybody to this presentation and update on the New Zealand Genetic Frontotemporal Dementia Study. My name is Sue Brewster and I'm the Executive Director of Auckland Medical Research Foundation. Our foundation has existed since 1955, and over those last nearly 70 years, we have funded over $96 million worth of medical research funding. So it's pretty spectacular, the results that we're starting to see from this. And Dr Brigid Ryan, who is at the forefront of this dementia study, we provided a fellowship to back in 2017. And this really enabled Brigid to work full-time on this particular study. And it actually first started in 2016 when the participants of a family came forward and asked to be part of this study, as it was found they had a mutant gene that made them have the possibility of getting this frontotemporal dementia. So the participants were recruited onto the study, and in 2018 the first parts of the study were started to be undertaken. So all sorts of tests and analysis on these family members, and to see if they could identify whether there were any early biomarkers or symptoms of dementia before the person actually showed real signs of having this frontotemporal dementia. It's fair to say the results have been spectacular, and it's going to lead the way in terms of, hopefully being able to have earlier interventions for dementia before it's actually diagnosed, and maybe even preventions. So I won't go into too much detail, but this is an update. Dr Brigid Ryan has been on maternity leave. She has had her first baby, and so we had someone else narrating through this presentation for you. But look, I hope you really enjoy it. It's phenomenal that we have been able to fund this type of work. So 2017 was when Dr Brigid Ryan got her postdoctoral fellowship through AMRF, and then Callagher Charitable Trust through AMRF were able to fund her in this ongoing work as well. So I really hope you enjoy the presentation. It's one of the world-leading studies that's going on at the moment, and I really look forward to any feedback you might have from this, which you can send to us at amrf@medicalresearch.org.nz. Thank you. Bye for now.
Welcome everyone, and many thanks for the opportunity to speak to you today. This is an updated report on a long-term project which aims to identify the early signs of dementia before symptoms appear, and is presented on behalf of Auckland Medical Research Foundation, Dr Brigid Ryan, and a multidisciplinary team of neuroscientists and clinicians from the University of Auckland and the University of Otago. This research is based on the premise that the key to improving the efficacy of current dementia medications and therapies, and ultimately prevention of the disease, is early detection. So what researchers are trying to do is identify the early signs of dementia before symptoms appear, so that treatments can start early, or in the long run the disease be prevented altogether. The brain changes that underlie dementia start many years before people notice symptoms. Trying to treat dementia once symptoms exist has so far proven unsuccessful. So the thinking behind this project is that early intervention, before symptoms appear, could be the key. In order to achieve that, two things are required. Firstly, development of treatments that work in the pre-symptomatic stage, and secondly, identification and observation of people who are in that early stage before they have symptoms. The research project we are reporting on today, the New Zealand Genetic Frontotemporal Dementia Study, is an interinstitutional, multidisciplinary project conducted by neuroscientists and clinicians from the University of Auckland and the University of Otago. Today we will discuss what has been achieved since the project was established in 2016. The work by this large team has been made possible with the help of the Auckland Medical Research Foundation. Their funding has been critical for this project, especially because it is a long-term project that requires ongoing support. So before we talk more about the study, here is a brief summary of frontotemporal dementia. Frontotemporal dementia, FTD, is a type of dementia like Alzheimer's disease. It gets its name because it affects two areas of the brain, the frontal lobe shown in blue and the temporal lobe shown in green. Damage to the frontal lobe causes extreme changes in personality. For example, people become disinhibited and behave irrationally, impulsively, and inappropriately. They often lose motivation and also lose empathy. Obsessive and repetitive behaviors are also common, for example, repeating the same phrase over and over, or obsessively walking for hours each day. Damage to the temporal lobe primarily causes language impairment, so people lose the ability to form sounds and say words, or to understand language or to remember the meaning of words. So people with FTD will have damage to one or both of these brain regions. FTD is divided into two subtypes, depending on the most prominent symptom, either behavior or language. Essentially, people living with FTD experience radical changes to their personality or their ability to communicate, which is devastating for both them and for their families. The main features of FTD that sets it apart from Alzheimer's disease are that it's usually young onset, meaning that people develop symptoms before the age of 65, and memory impairment is not the first or most prominent symptom. FTD is also much more likely to be inherited than Alzheimer's disease. In about a third of cases, it is caused by an inherited gene mutation. In our study, the affected gene is called microtubule-associated protein tau, or tau for short. Mutations in this gene cause the tau protein to clump together and damage brain cells. You may have heard of tau because it's one of the proteins that is affected in Alzheimer's disease. So how many New Zealanders are affected by FTD? It's not very common in older people with dementia, but it is one of the most common causes of young-onset dementia. We think that about 2,000 people in New Zealand are affected by FTD. The New Zealand Genetic Frontotemporal Dementia Study has its origins in a single donation to the Neurological Foundation Human Brain Bank. In 2006, one member of this family passed away and her brain was donated. Subsequent genetic testing revealed that the woman carried a dementia-causing gene mutation, and through that donation, the family became interested in research, and they actually approached researchers at the Centre for Brain Research to ask if they could be involved. That's quite an unusual way for a study to start, and very unique in having a family cohort of this size involved in real-time dementia research. So the researchers are testing and identifying people in this family who carry the genetic mutation that means they will develop FTD in the future. If a parent has FTD, the person has a 50-50 chance of inheriting the mutation and developing FTD. By comparing people in this family who carry the mutation with people in this family who don't carry the mutation, the researchers can identify the very earliest changes caused by the mutation. This project was really initiated by the family themselves and has been a collaborative process with the family all the way through. And because this is being done in a research capacity rather than a clinical capacity, the information about mutation status is not being given to the participants. But if they want to find out this information, they are supported to go through the clinical process so that they can be informed. Some people in the family do want to know if they carry this mutation, so they can plan for their future. Some people don't want to know. The participants' age range starts from 25, which is the youngest in the study. And that's because that age is the earliest that researchers think changes might start occurring, if the participant has the mutant gene. That equates to around about 30 years before symptoms might be expected to show in this family. And previous research suggests that it is likely that undetected changes start to happen at that early stage. This is a common feature of many types of dementia, that these changes in the brain happen a long time before symptoms are actually apparent in a person. And so this represents a really amazing opportunity to intervene during the pre-symptomatic phase, the window of change where there is something starting to change in the brain, but it hasn't got to the point yet where it's causing symptoms. Here is the family tree of those being studied. We are only presenting a partial family tree for privacy reasons, but this is a large family. The black circle or square indicates people who have been affected by frontotemporal dementia, and the line through the symbols indicates people who have passed away. The family tree shows that people have been affected by FTD through many generations of this family. Affected participants in this study typically demonstrate behavioral variant FTD, which is characterized by a progressive deterioration in personality, social conduct, and cognition. The exact characteristics vary between participants, but the most common presenting features are antisocial behavior as a result of disinhibition. For example, sexually inappropriate behavior, trespassing, and aggression, to the extent that there has been police involvement in 2 cases. Obsessive behaviors are also displayed by the affected participants, presenting as things like walking for many hours a day. As mentioned before, FTD is generally a younger onset dementia, and onset in this family varies between mid-40s and mid-60s. In keeping with similar genetic FTD cohorts, about half of the participating family members exhibit Parkinsonism. As the disease progresses, individuals become incontinent, mute, and immobile. Survival varies widely between individuals, from between 5 to 21 years after diagnosis. Interestingly, one of the family members presented clinically with typical Alzheimer's disease, so even though they carried exactly the same mutation, they met the clinical criteria for Alzheimer's disease rather than FTD. In addition to the original donor, one other brain from this family has been donated to the brain bank. The neuropathology in these individuals is typical of FTD, with temporal and frontal atrophy, mostly symmetrical. This image is a coronal T2-weighted MRI from one of the family members, so the white areas show severe symmetrical temporal atrophy. The 2 temples on the sides of the brain have shrunk significantly, consistent with FTD. The tau pathology in this family cohort is caused by a mutation in the tau gene. FTD is genetic in about 1/3 of cases, and tau is one of the 3 main genes that's involved. There are at least 63 mutations in tau that cause FTD, and this particular mutation is one of the most common, being reported in 48 families previously, who are all likely to have a common Welsh ancestor. The original donor's father emigrated from Staffordshire to New Zealand as a child in 1923, so it's likely that this family is directly related to the similar cohorts that have been described in the UK. As with other known FTD mutations, anybody who carries the mutation will develop FTD. Turning now to the procedural aspects of the New Zealand Genetic Frontotemporal Dementia Study, its aim is to study individuals who carry this mutation, but do not yet have symptoms. So, what has been done so far? The first step was to recruit participants into the study and determine whether they were pre-symptomatic mutation carriers or non-carrier controls. The researchers started recruiting in 2016, and it took around 2 years for all participants to have their first assessments. Inclusion criteria include being at a minimum age of 25 and not having any FTD symptoms. The researchers have now completed genetic testing for all participants. The aim of the testing has been to identify pre-symptomatic biomarkers in this cohort. Essentially, as many potential biomarkers as possible are being measured and compared between mutation carriers and non-carrier controls to identify any biomarkers that might indicate early disease processes or predict disease trajectory or how they respond to treatment. Participants come in annually for blood samples, tests of their sense of smell, and a comprehensive neuropsychology assessment battery. The researchers are specifically interested in olfaction, the participants' sense of smell, because it's affected early in other neurodegenerative diseases, such as Parkinson's disease and Alzheimer's disease. In addition to these annual measures, researchers also conduct a number of sub-studies including brain imaging, retinal changes, social cognition and memory, and most recently hearing and balance. These are all measures that have the potential to be useful as very early biomarkers of dementia, so basically as many different markers as possible are being tested. It's really important to do this testing over a long period of time so that anything that is changing can be seen as people get closer to developing symptoms. This study is observational because the family is not being given any interventions. Researchers are just observing what's happening to them over the course of their lives, and what they're doing is looking for changes between the 2 groups in the family. So, one group is people in the family who will go on to develop dementia, carriers of the gene mutation, and the second group is people in the family who won't go on to develop dementia, non-carrier controls. Ultimately, researchers hope that any changes identified will be able to be used to develop a test that can then be used in the general population to identify people who are at risk of getting dementia at some time in the future and would allow for interventions before those people develop symptoms. So here are some highlights from the results seen so far. The retinal study is led by optometrist Hannah Kirsten. Hannah has used optical coherence tomography, which is an imaging technique used to obtain cross-sectional images of the retina. So in image A, you can see a photo of the retina, and in image H, you can see a cross-section of the retina with the retinal nerve fiber layer segmented into its various components. Hannah has used these images to measure the thickness of many different regions of the retina. She has used a related technique, OCT angiography, to visualize the blood vessels in the retina. Hannah observed many components of the retina using these measures, but did not find any difference between the pre-symptomatic carriers and non-carrier controls at their first annual visit. These assessments will be repeated in future analyses to see if any changes can be identified over time. Researchers have also analyzed baseline data for cognitive deficits. This work was led by Ashley Baker during her PhD, who was supervised by Lynette Tippett and Donna Rose Addis. Ashley and the team's clinical neuropsychologist Christina Ilse performed all of the testing. Ashley has looked at many different measures of cognition and found deficits across many cognitive domains, even in the younger group who are up to 30 years prior to expected onset. This figure shows 2 tests of language, and the results indicate that the mutation carriers, shown as dots, perform worse than age-matched non-carrier controls. Tests have also identified a decrease in the sense of smell, even when mutation carriers are still in their 30s. This figure shows the change in sense of smell over 2 years. If the person's sense of smell improved, their score would be in the top half of the figure. If it worsened, their score will be in the bottom half of the figure. So you can see that all of the mutation carriers shown in red have had a decrease in their sense of smell over time. So the collective data to date indicates that, as predicted, it is possible to pick up very early changes in people who carry the gene mutation, even decades before they are expected to develop symptoms. This is giving the researchers confidence that their rationale is sound and that there is hope for preventative treatments. So what's the point of this work? What impact will the New Zealand genetic frontotemporal dementia study have on the health of New Zealanders? It's possible that if early intervention is successful, these treatments could be used preventatively in patients with genetic and non-genetic FTD. And these biomarkers may be relevant to other tau-related conditions, including Alzheimer's disease. So that's why this work is so vital, given the predicted increases in all types of dementia and the critical need for effective preventative interventions. The researchers hope that, with the ongoing support of funders like the Auckland Medical Research Foundation, New Zealand researchers can continue this world-leading work and contribute to the global effort to prevent or treat dementia. The next step is to collaborate with international studies to validate biomarkers of pre-symptomatic FTD that have been identified in this study. Such biomarkers are critical to identify participants for clinical trials of new treatments. The New Zealand FTD study researchers have also established collaborations with major international studies of genetic FTD, which are doing similar work but on a much larger scale across multiple countries. Researchers will work with these studies to share data and validate the New Zealand findings. The group has been invited to take part in the FTD Prevention Initiative, which is an international consortium of genetic FTD studies that aims to work with pharmaceutical companies to design clinical trials of potential treatments. With less common diseases such as FTD, international collaboration is critical to recruit sufficient participants for clinical trials. So the New Zealand involvement, even with a small number of participants, will be important. We hope that this presentation has helped to instate hope and build confidence that this work into early detection of dementia is fundamental to the development of groundbreaking prevention and treatment strategies that don't currently exist. We would like to thank the participating family and Dr. Brigid Ryan, along with the research team, for their unwavering commitment to this study, and also Auckland Medical Research Foundation and their donors for funding research into a neurological disease that impacts so many. Finally, we would like to give a very special thanks to all of our supporters, people just like you, whose donations to the Auckland Medical Research Foundation enable these dedicated researchers to continue their life-changing work. If you are not already a supporter, we encourage you to make your donation to the Auckland Medical Research Foundation and support our future today. Simply go to the address on screen, medicalresearch.org.nz, make a donation.